do Departamento de Ciências Atmosféricas. Então vamos voltar no boa tarde a todos. Hoje nós temos o seminário do Departamento. Não contou até 5?
Tem que contar até 5?
Vou repetir, hein? É assim, nós estamos no seminário, nós temos a presença hoje da Agnès Borbon, a professora Dalgisa vai apresentá-la. Tem bastante gente hoje aqui no auditório, tem bastante gente hoje também no meeting. A Agnès, eu gostaria de agradecê-la pela disponibilidade de apresentar o seminário e vamos às apresentações que a professora Dalgisa vai fazer.
Bom, gente, agradeço a presença de todos e o pessoal que também está online. E agradeço a professora Agnès Borbon. Sorry for the people for French, but I will present Agnès em português. Bom, eu acho que é interessante falar um pouquinho, ela pediu para eu falar uma longa história sobre o projeto BIOMASP, mas o mais importante é a longa história com a professora Agnès, que, antes de tudo, começa com a Pamela Dominucci, que fez o doutorado comigo, e a Pamela foi para um evento na Europa, e ela recebeu um bilhete no pôster de alguém querendo conhecer o nosso projeto. E aí começou a nossa história, isso foi em 2013, 2014. Depois disso, em 2015, a professora Agnès, nós tivemos um projeto CAPS PVE, né, professor visitante especial. A Agnès esteve aqui durante um mês, trabalhando com a gente. Nesse período, logo em seguida, a gente teve um FAPESP, um sprint, onde nós criamos o nome BIOMASP, junto com a professora Silvia Ribeiro de Souza, que agora, antes era o Instituto de Botânica, agora é o Instituto de Pesquisas Ambientais. E nós começamos, nesse momento, a trabalhar nas áreas, né, de Mata Atlântica remanescentes aqui em São Paulo. Começamos a nossa história no Morro Grande, aqui na USP, e também lá no Instituto de Botânica, região do Parque Cientec. A professora Agnès, ela é formada em Química da Atmosfera, seu doutorado em 2002. E desde 2005 ela trabalha no CNRS da França, e com mais de 70 artigos publicados, é uma pessoa bastante atuante na área de química da atmosfera, envolvendo compostos orgânicos voláteis. Agnès, muito obrigada, e muito obrigada a todos.
Muito obrigada, Gisa. Sim, estou feliz de estar aqui com vocês. Agora vou falar inglês. Okay, people, so thank you very much for being here. Thank you, Gisa, for the introduction. So today I'm here in the frame of a 3-month visit I got from FAPESP to carry out the intensive field experiment we have here at USP and in Morro Grande in the frame of the BIOMASP Plus project. So today my purpose is first to tell you a little bit about trees and urban atmospheric pollution, to move forward the BIOMASP Plus project. I will show you what are the objectives of the project, what type of strategy we implement, especially on the field, and I will also give you some preliminary flavor of our first results. So yes, I'm a scientist at CNRS, so I don't know if you know CNRS, but it's a national center for scientific research in France, and I work in a laboratory located in the central of France called LAMP. So LAMP stands for Laboratoire de Météorologie Physique. So maybe you picked up the word météorologie, yeah. And so most of the people there are physicists, but there are some chemists like me, and more chemistry is performed in this lab. So to start with, so now I know about, okay, next slide. So to start with, I would like to stress that forests are benefits for us, especially in a global change context. So I picked up here some very nice pictures of the vegetation you have the chance to be surrounded by here on the campus with different trees. And first of all, I would like to say that forests are beneficial for our health and well-being for all of us. And just an example, here we had the chance here at Bourg-Granger, oops, sorry. Okay, yes. To have these araucaria trees, and we also had the opportunity to taste piñaus, and I guarantee you that it was very good for mental health on the field, which is very intense. Forests are benefits for health, human health, but not only human health. They are also benefits for many aspects. So I focus here on different aspects: the managing of stormwater, the increasing of neighborhood property values, the filtering of pollution as well, the cooling, especially in a warming global climate. Oops, I think now I don't get the pointer anymore. Okay, it works. Reducing as well rates, so related to cardiovascular diseases, so related to the healthy of people. The protection of biodiversity, as well as reducing obesity levels. Just a quick overview of the benefits of forests. But we can also look at forests from a different point of view. I pick up this sentence in a book Giza gave me, and I will give it back to you when I'm coming back to France. A book describing the fauna and flora on the campus. And I like this sentence because I think it summarizes the idea I would like to develop after. Because forest is also an island within the sea of a man-made perturbation. And behind this sentence, I think there are two ideas. The first one is that the surface of our forest is getting little and little. And I think here, from the Mata Atlantica perspective, only 7% of the forest remains. And as well, behind man-made perturbation, we know that anthropogenic activities emit in the atmosphere different pollutants, both gaseous compounds as well as particles. And these particles will also interact directly with the flora in presence. Oh my God, I'm not so. So first of all, urban pollution can affect the plant metabolism. So this is some results from a colleague here who is a member of the project, Claudia Fernal, where she looked at the ozone-induced injury on plants which are submitted to ozone fumigation. So here you can see that the ozone effects after the exposition to ozone are quite visible, where you see here this upper surface stippling. Usually the effect of ozone on a leaf is regarded through the leaf injury index, which is shown to be correlated to ozone accumulation in chamber experiments. So the plant response to ozone are illustrated through the modification of their stomatal conductance, their photosynthetic rate, their transpiration rate, as well as their secondary metabolism. Just keep in mind that plants are sensitive to ozone exposition. Urban pollution also affects plant emission. So plants produce in the atmosphere some gaseous compounds. These compounds are known as biogenic volatile organic compounds under the BIOC label, and these compounds are emitted through different biosynthetic pathways. So the emission of BVOC by plant is an illustration of their defense against biotic and abiotic stress, as well as they are also useful for their reproduction. And this emission are performed through gas exchange between the stomata and the atmosphere. So here I give you an illustration of how pollution, and especially the exposure to ozone, will modify these BIOgenic emissions. So here usually we make the difference between BIOgenic emissions, which are constitutive of the plants when the plant is not submitted to a stress, versus the BIOgenic emissions which are induced by a stress. Okay? So usually these BIOgenic emissions, which represent a very large spectrum of compounds, are categorized in 4 groups. BZ is for benzenoid, it's for further understand than the figure. SQT is for sesquiterpenes. GLV are green leaf volatiles, and MT are for monoterpenes. So here it's, you see the results from a study in Beijing Megacity, where they looked at the modification of the BIOgenic emissions categorized in this 4 groups before an exposure to ozone, and after an exposure to ozone. I'm sorry, I'm not angling with the pointer. Okay, it's better. And so you see that, first of all, after an exposure to ozone, we see a clear increase of the BIOgenic emissions, so it affects the intensity of the emission, as well as it modifies the relative proportion of the different group of compounds. And on the x-axis, you have the different types of trees they studied. So keep in mind that plants emit BIOgenic emissions, and this BIOgenic, when they can be stress-induced, and these emissions are modified compared to non-stress plants. So, through the emissions of BIOgenic, vegetation will affect urban pollution. Why? Because BIOgenic emissions are very reactive compounds for the atmosphere. So they are known especially as ozone and secondary organic aerosol, SOA precursors. So I give you here 2 examples. First of one is the MEIR index, maximum incremental reactivity index, which gives you the ability of a volatile organic compound to produce ozone. So this index was established by Carter some years ago. So here I reported the MEIR index of different anthropogenic VOC, which means that VOCs emitted by anthropogenic activities. You see different VOCs here reported. And when we report the MEIR index of some key BIOgenic, isoprene, and pinene here, you see that on this scale, these BIOgenic are as significant as other anthropogenic VOCs to produce ozone. Regarding the ability of BIOgenic to produce secondary organic aerosol, I reported here the SOA yield of different compounds. So in blue and gray, you have anthropogenic volatile organic compounds, and here inside the green square you see some SOA yields of BIOgenic, and you see that the SOA yield of this BIOgenic are higher than the one of commonly or let's say traditional anthropogenic VOCs. So these compounds are highly reactive even compared to more traditional anthropogenic VOCs. So these biogenic VOCs make a large component of gaseous pollution. In particular, they contribute to the ozone NOx and peroxy radical cycle. So I won't go into details into these cycles, just to keep in mind that ozone is produced through a cycle called the Leighton cycle. Oh, sorry, the Leighton cycle. And when we add some volatile organic compounds, and BIOgenic in particular, represented here in green. This BIOgenic will modify this cycle, especially by let's say, suppressing the conversion of NO to NO2, and by producing ROX. The production of ozone will depend on the availability on both NOx here through NO and NO2, as well as volatile organic compound. And this availability is usually expressed as a ratio between VOC and NOx. Depending on the value of this ratio, and depending on the region where we are, the production of NOx will be either sensitive to the presence of VOC or to the presence of NOx. This is what is illustrated here. So here we have, let's say, a commonly admitted scheme regarding ozone production depending on the availability of NOx and in black, and volatile organic compounds here in green. So here on the x-axis you have the different area, the urban area, the suburb, and the rural area. So as far as we go further from the urban area towards the rural area, usually we see a decrease of NOx, NOx being mainly emitted by traffic emissions. I'm not very good with this pointer. And at the same time, the volatile organic compounds increase because with the increased presence of biogenic volatile organic compounds. And so usually we define two regimes depending on the ratio between VOC and NOx. The first regime corresponds to a ratio lower than 4, when we know that ozone is forming in a low NOx regime, and another regime where ozone is produced in a high VOC regime, which is usually NOx limited. And now, in the context of the reduction of NOx emission with the introduction, for instance, of clean technology on car, for instance, we can expect that this ratio and the frontier between those two regimes will shift a little bit towards the urban area. So we expect in the future, and this is represented here on the bottom graph. I'm sorry, I'm not very good with these things. We will extend the area where the BIOgenic emission in particular will play a key role. So these organic compounds also make a large component of aerosols. So I switched to aerosol after the gaseous phase. So usually the organic aerosol is made of two components. A primary component, which is here illustrated by the term POA, primary organic aerosol, and the secondary component, the SOA, I told you about before. So usually it is admitted that secondary organic aerosol makes the larger fraction of the organic aerosol mass, usually higher than 50%. So this is extracted from the results from Jimenez et al. In 2009, who made a kind of overview of the OA components globally. Now, there are some recent studies that show that the primary organic aerosol component could be more important than we already thought. So here I illustrated some results from a French study in 28 French cities where they sample organic aerosol on a high volume sample filter and analyze their composition with a large spectrum of organic markers. And here they represent, on the right hand box plot, the ratio between the primary organic aerosol versus the total organic mass. And what we can see here, so first we have a large spectrum of fraction from primary organic aerosol, but still this contribution of POA to the PM10 mass is quite significant. It can be up to 50% here maximum. So this is a large component which so far has not been so well considered in urban areas. More or less, there is also a complexity when we think about an urban area with the mixing of these biogenic components and the anthropogenic emissions. So two illustrations here. The first one shows the role of nitrogen oxide on the biogenic formation of secondary organic aerosol. So this is an illustration from an aircraft campaign where they explored the Manaus plum while the Manaus plum was traveling over the Amazonian forest, and they represent here the observation here of the concentration of the organic aerosol, which are represented in orange. And they also model this organic aerosol, represented here in blue. So each peak corresponds to a cross-section of the Manaus plume. And then so what you see is that the model is doing a quite good job. But what they do is at some point in the model, they switch off the anthropogenic emissions. And this is the result here from the green curve, showing that if we switch off the anthropogenic emission, we have an important underestimation of what organic aerosol should be. So this is to show you that it is very important, even when we are interested in biogenic SOA formation, to also consider this anthropogenic fraction. The main reason for that is that in the presence of NOx, we have different secondary organic aerosol route formation, and I show here the different oxidation route related to isoprene, depending to the NO availability here, which is represented here by the arrow. So as soon as we go, as long as we go up, there is an increase in NO. And what you can see is that depending on the availability of NO, we can find different routes that would lead to the formation of secondary organic aerosol with different compounds and, as a consequence, different properties. So given this context, given the importance of biosphere-atmospheric pollution interaction and their complexity, we decided to launch this BIOMASP project, which stands for Biogenic Emissions, Chemistry and Impact on the Metropolitan Area of São Paulo. So our objective is to better understand the role of this biosphere-atmosphere interaction on urban pollution. We first by tackling their physico-chemical as well as its biological dimension, and in a biogenic emission and atmospheric perspective. So Giza already said some words about the history of the project, so I won't repeat, but you all understood that it started with Pamela Dominuti, and Pamela is still a part of the project 7 years after. Just some reminders here, at least for Giza and myself. So my visit started in 2015. Then Giza and Sylvia, you visited us after 2018, after we launched the first BIOMASP bilateral exchange. And this project allowed us to perform the first measurements of BIVOC in MASP. And then in 2020, we submitted a bilateral project to our both agency, FAPESP and ANR in France, which was accepted. And the project started in 2021 and we end in 2026. And nowadays, the project, mainly the agencies funded consumables, postdocs, PhD student grants, at least from your side here, and let's say all the experiment efforts and modeling efforts which will be performed But so far now we miss some support for people mobility, because in such a project it is very important that we pay attention to that the work is performed in a collaborative way. So now we are working hard on the coffee cube call, which has to be submitted very soon. Okay, so just here the people who are involved in the project. So first of all, this is a multidisciplinary project, so which gathers chemists, atmospheric chemists, physicists, meteorologists, ecophysiologists, and biologists. Okay, so the number of partners is 9, and since then we have new partners here. In France we have the LSCU and INERIS, we have here the Institute of Physics at USP, as well as IPEN, so I think which it's very good. And after this mission we were also quite successful with getting postdocs and PhD students to work with us. So why São Paulo? So São Paulo is our domain of investigation. São Paulo is a subtropical emblematic megacity for us because of its high levels of pollutions. And especially if we think about ozone, at some point there are some decreasing trends of ozone, but not systematic, and at some station we also see some increasing trend. And there is also the question of how these biogenic volatile organic compounds could play a role in those trends. And it also surrounded by the Mata Atlântica forest, which represents 30% of the metropolitan area. Its climate is favorable to biogenic emissions and strong photochemistry. And we had the chance to have, before submitted BIOMASP Plus, this bilateral project, which allowed us to make the first BIVOC screening all over the metropolitan area, with a selection of 3 sites, which are represented here. So PEFI, which is located at the Botanical Institute, USP Matao here, and the reservoir of Morro Grande, 40 kilometers southwestern part of the metropolitan area. So during this project we also get an idea of the air mass circulation as well, with mostly southern air masses, which make us confident with the strategy we wanted to deploy, with, let's say, an urban site in the metropolitan area, but which is potentially impacted by the biogenic emission from the forest located in its southern part, and a forest supersite also influenced by biogenic emission, but quite close to the metropolitan area. And another point I wanted to stress here, I told you about previously about the importance of primary organic aerosol, and there are also some studies here from IAG showing that the primary biogenic component of PM10 MASP from a long-term survey was also a significant component of PM10. So this is a schematic concept of the project. I won't go in detail through it, but this is, for me, the opportunity to show you about the strategy and how the project is organized. So we define 4 tasks in the project. So the first task is to quantify the BIVOC emission from the Mata Atlântica forest, which is a forest which is so poorly known in terms of biogenic emissions, as well as address its response to urban stress. We also have a second task related to the investigation of the atmospheric composition in terms of gas and particles, in order to better address the processes related to the biogenic anthropogenic mixing. And for sure, to relate both, we need a good understanding of the meteorology through the characterization and quantification of fluxes and dynamics. And then we have a modeling task which will integrate all the knowledge we will gather from these 3 other tasks through, in particular, a modeling task with the WRF-Chem model coupled to the biogenic emission model MEGAN. So now let's go deeply into our strategy, observational strategy, which mobilizes a lot nowadays. So we have two, oops, no, how do I go back? Oh, it's okay. So we have two contrasted sites, the primary forest located at the Réservoir Morro-Grande, as well as the urban forest with a measuring platform here at IAG. So here I plotted again the wind roses we get here usually. So to implement those two contrasted supersites, we need to build a new infrastructure. And here I take the opportunity to congratulate my Brazilian colleague about all the hard efforts they made to make this infrastructure. Bon? Yes. Okay. Okay, so at the two sites we duplicate the experiment observation. So we have some activities related to the characterization of BIVOC emissions, activity related to eddy covariance and relaxed eddy accumulation flux measurements, activities related to the characterization of particulate physics, chemistry, and microbiology, as well as activities related to the online organic speciation of VOCs and organic aerosols. So now I will go through the strategy we deploy. So to start I start with the biosphere. So you see here the identification of one of the trees that was selected in Morro Grande on the picture. So first of all, the aim is to quantify BIVOC emission at the leaf scale. So the first step of this activity was to perform a characterization of the flora in presence and to identify the dominant species on each site. So here you have a list of the species that were selected in Morro Grande, and the same work will be performed here at USP. Once the species are selected, they are cut, they are put in a water basin and transferred to the lab, where then they can be analyzed. So their biogenic emission can be analyzed at the laboratory through the sampling of VOC on sorbent tube and their analysis by GCMS. So GCMS stands for gas chromatography mass spectrometry. So you see here the instruments. Oh, yes, you're right, Giza. It's okay, nobody will see.
No problem.
So the focus was on the leaf scale, but after some recent discussion, we also decided to look at other compartments of the forest by also quantifying the emissions of the stem. Here you see an example of the setup. The litter as well, with the deployment of this glass chamber, as well as the water, because in Réservoir Mont-Rogranger we have this big lake quite close to the sites, and this lake can potentially act as a sink or source of BIOC, as well as also a source of latent heat fluxes. So this is also a major question we have. So first we quantify BIOC emissions from the different compartments, and there is another activity related to the evaluation of the plant's response to abiotic stress, in particular ozone exposure and drought. So this is performed this time in the laboratory. So you see here an example of an experiment which was performed by a French PhD student who visited Sylvia Ribeirzo's lab at the end of last year. This is here the plant she works on, and she performed ozone fumigation in this chamber here, where the plants are exposed to 80 ppb of ozone, and then perform the VOC quantification with the same setup as I showed before. And here is the flavor of the preliminary results. So the different colors corresponds to different experiments between controlled experiments where there is no ozone, and different experiments where the plants are exposed to ozone or have been also submitted to drought. And what is interesting here, if we keep in mind that one peak corresponds to one biogenic volatile organic compound, is that depending on the experiment, especially the one looking at stress compared to the control, we have different peaks and different signature, which shows that we have different BIOC signature depending on the type of stress. Now we go back to the atmosphere. So when we want to look at the interaction between biogenic emissions and the atmosphere in anthropogenic influence environment, one way to carry out is to work at the laboratory. And here this is an experiment we plan to do in the 2 coming weeks, and even in the 4-5 coming weeks, where we will go in at Sylvia's Ribeiro's lab as well, and we work with the same way as we did before to quantify VOC emissions, but we will connect to this chamber what we call a PAM reactor, which is a reactor when we can simulate the photochemistry in the atmosphere, but in a faster way. And after this reactor, you have different instruments, the same as we deploy on the field, that will especially allow us to quantify the potential of these biogenic emissions to form secondary organic aerosols. So it's another way to look at the reactivity potential of those biogenic emissions in a controlled environment. But most of the effort for us is towards the field. So here I wanted to show you the 2 different infrastructure we have at the 2 sites, starting with Mont-Rogranger. So this is here the tower. So along the tower and at the top we have different micrometeorological sensors. And at the bottom of the tower we install this laboratory with different instruments which allow us to characterize the atmospheric composition of gases and particles. So I won't go into detail into the different instruments we have, but this is a quite full space. Close to the tower, around let's say 200 meters, you see here the map of the reservoir. The tower is on the western part and this trailer is here. We have also some measurements that are performed which will complement the measurements we do at the bottom of the tower, thanks to the trailer deployed by the Institute of Physics here at USP. And close to this trailer we also have a high volume sampler to collect filter, the same way as we do here at RMG. And here just a picture of one of the samples we collected maybe 4 or 5 days ago. And you see that after 12 hours, it is quite clear and almost dark, the material we managed to collect. So now we go back here at IAG. So again, we have the tower here with the different micrometeorological sensor. And at the bottom of the tower, we have different laboratories, one hosting the SMPS for the aerosol distribution, and an attelometer for black carbon measurements, as well at the same time of instruments that were shown before for the Réservoir Moraux-Granger. So a big technical and instrumental setup going on. And on the field, as well in Moraux-Granger, we also deploy, so quite soon, maybe today or tomorrow, the PAM reactor to also look at an accelerated photochemistry to look at the reactivity potential of the biogenic air masses inside the canopy, but at least with a strong anthropogenic signature, which I will show you later. Okay, so now I will finish with some flavors about our atmospheric results here. So just to tell you that during this intensive campaign, which I think started at the end of March, we had weekly meetings online with people from France and Brazil, some at Moraux-Granger, some here, some at home. And we also publish weekly bulletins summarizing all the activities going on, as well as we had the chance to have a regular forecast made by Karine Gama, who I think who was trained here, who is a meteorological forecaster, and Karine provides the forecast at least 2-3 times a week. So first of all, first flavor of the meteorology at ground level at both sites. So here you have the time series of the air temperature during the campaign. IAG is in blue, RMG is in red. And the relative humidity. So we clearly see the expected cycle we have for the temperature. Here, after May 9th, we clearly see the arrival of the cold front with a decreasing of the temperature and an increase of the relative humidity. On the right panels, you have the wind roses, confirming that at both sides the dominant wind comes from the south sector. And also an interesting thing we discussed yesterday during our meeting is the difference in temperature between both sides. Usually at RMG the temperature is lower, especially at night, and this may probably be an effect of the urban heat island we have here at IAG. So now let's go to atmospheric composition. So with the first highlight on the ozone time series. So this is the ozone time series at Réservoir Moraux-Granger. This corresponds to the red and orange curves because ozone is both measured at the trailer and at the bottom of at the top of the tower, sorry. And what we see that we have clear, consistent cycles, and we also see the typical diurnal cycle at the station. And then also superimposed the ozone observation we have here at IPEN, and we see that the well superimposition of the time series, showing also the large-scale regional signature of ozone. We got significant level during this period, especially at the beginning of the campaign, with level up to 100 ppb. And I think that's it for ozone. Another secondary pollutant here we measured at the tower is formaldehyde. So formaldehyde is a volatile organic compound. It's mainly a secondary product of other primary VOC oxidation, especially from isoprene. So I just made a focus on what we call our golden days, where we got this high ozone concentration, the highest temperature. And you clearly see that the formaldehyde time series here in blue well follows the one of ozone. So pay attention here, this is not concentration, it's just a normalized concentration because it has not been quantified yet. Now I switch to the gaseous precursors of ozone and SOA with a first focus on the traditional BIVOC, isoprene. And I've plotted here its journal variability at the RMG site, here on this graph. At RMG, we have two instruments measuring isoprene, a PTR of MS and a GCFID, and it's important to be sure that both instruments show the same trends and levels. So here in the plain line corresponds to the GCFID measurements and the dots corresponds to the PTRS, and we see that we have good consistency between both, and we clearly show the typical journal cycle of isoprene, mostly related to its biogenic emissions, which are temperature here and power dependent, light dependent. So we have consistency between isoprene variability and its controlling factors here. Now if we go back to isoprene measured here at IAG, so here I plotted the time series all along the period. Isoprene is in green. So we also pick up this journal cycle, and we also plotted with OLA one of the major oxidation products of isoprene, which are metachlorine and methylvinyl cetone, and we also see a clear consistency between these products and their primary precursor, but with a little shift in time because we need time to allow the chemistry to develop. And the highest isoprene concentration are quite consistent with the different ozone period we have. So the highest isoprene we have corresponds to the high ozone level, the highest temperature, and the lowest level corresponds to the low ozone period associated to the coldest temperature. So all what we've spared is what we could as well expect. And we also compare the time series of isoprene, of the oxidation products at both sites. And we see that at RMG, the levels of the isoprene oxidation products, at least for this period, it needs to confirm on the longer term, are quite higher, which maybe implies some higher oxidation at the forested site. After a BIVOC, now I switch to benzene, which is an anthropogenic VOCs. Again, this is a time series at IAG. You can see that the highest benzene also corresponds to the higher pollution-loaded periods. And what is here surprising is that we also superimpose both time series for toluene at both sites, and surprisingly we see higher level here at this period at the forested site, which was for me quite unexpected. So we were wondering about the effect of local emissions, and at the site we clearly see some local emission, especially anthropogenic emission effects, and also the role of dynamical transport there. And to finish with, here is the time series of the PN1 composition seen by the aerosol mass spectrometer that were both deployed at both sites. So on the upper panel you have Morogrange, here you have IAG. So this time series shows you the organic sulfate, ammonium, nitrate and chloride composition. So we see the clear dominance of organics with some difference in trends at both sites. And during the last days here, we clearly see strong organic aerosol levels at IAG, much more moderated here at RMG. And we started to perform a first positive matrix factorization in order to deconvolute the different oxygenated states of this aerosol. So here in orange, in red, you see the secondary organic aerosol component, which is quite elevated at Morogrange. But we also see a signature of traffic at this site. And that's it for now. So some points I wanted to stress here. So we made a strong and unprecedented experimental effort to tackle the biosphere-urban air pollution interaction here in the metropolitan area of São Paulo. The campaign is successful at both sites, and we have promising results with a recovery of the measurements higher than 80%. We got some contrasted situation with low and high photochemical production. We also see anthropogenic signature at the Morogrange site. We still have some upcoming experiments going on. I won't go into detail. And now it's time for data analysis and joint comparison. And as I told you before, we are working especially on this coffee cup call for mobility. And to finish with, I'd like to address or to thank all the people involved in the success of this field experiment. People involved in shipment, because shipment from France to Brazil is a big deal. Logistic, payment, meteorological forecast. All the people now involved in the field. Congratulations to all of you. And I want to finish with special thanks for the people who makes us feeling good. The Tudobem people, the commuting driver, Valmir, Sergio, our Uber driver, and others, and our hosts, especially Felipe, José, and I address a special thanks to Giza and Mauro and Jordi Murizo. Yes. Thank you. I hope it was too long. Obrigada. And I think this with a sagui.
Hello. Well, thank you, Agnès. Perguntas, questões, sugestões, dúvidas?
Dúvidas is doubts?
Está aberto aqui também, pessoal. Se alguém quiser fazer alguma pergunta, eu não consigo ler, mas alguém lê para mim, porque o Richard lê para nós, né, Richard? Ninguém quer fazer nenhum comentário? Se ninguém for perguntar, vou perguntar.
I have a question.
Fábio, pode falar, please.
Yes, since the doctor is 3:30. Just a curiosity, the concentration, toluene and benzene concentrations are harmful for us human beings because it's really high. I didn't expect that. For me, it's new that the ozone stress plants to produce such cancerogenous VOCs.
You mean the toluene concentration here?
Or benzene, yeah, both.
No, so benzene is not so, well, for me what is surprising is the high level of toluene at RMG.
Yeah.
But we have to catch, and it's just a 3 days period, but still it's important. So usually when I'm thinking about toluene, I think about its anthropogenic origin, but there are some papers showing that toluene can be also emitted by stress plants. There's some work about it. So this was a question I have. So far I don't have an answer, but this is maybe something we need to investigate, and maybe we also need to put this level in the perspective of the long-term measurements.
Yeah, anyway, they can be harmful to us, our health, because they are cancerogenous.
Benzene is the one who is regulated. Other aromatics, they are regulated in outdoor and work environments, but not in the atmosphere. The ambient level in the atmosphere is not so far regulated.
Okay, that's good to hear.
Yeah.
Ok. Rita?
Congratulations to you all. It's really hard work. Agnès, why is there a peak of isoprene early in the morning?
So isoprene usually is considered as a biogenic compound, but in urban area, and it was part of my PhD work, we showed that isoprene has also an anthropogenic component. It can be emitted by traffic as well. I remember it was in northern France. We showed that in summertime it could be a third of the isoprene level. So yes, isoprene is also emitted by traffic. So I think well, this is the journal variability I showed here. We can get some anthropogenic isoprene at Moraux-Granger, but we have to pay attention because, the problem is also the time scale. So for PTRMS so far, I cannot say if we see this peak. But for the GCFID, there is also an issue because the GCFID is a specific instrument, which means that one peak should correspond to one compound. But we can have some what we call coelutions, meaning that two compounds can be released at the same retention time. So there were some doubts about some coelution effects, if I remember well, with hexane at the site. So I think this still needs to be validated by the data analysis and all the validation steps now we have. So far for now, I won't say anything or made any assumption of these peaks before we validate the data. But this was an issue of Adrien Gandolfo, who was in charge of the deployment of this instrument. But we have anthropogenic isoprene, but maybe not at Moraux-Granger. So far, I cannot say anything about it.
The Tallini result?
Yes, there is also Tallini. You're right, Shiza. Yes. You also quantified this fraction. So for sure, it's a flavor of some of the measurements we did, but there are many others I didn't show.
Alguém mais aqui no meeting? Ninguém aí está interessado? Pode falar direto, não precisa escrever. Nada. Tem alguma coisa no YouTube?
Não, só o Francisco Rodrigues agradeceu pelo compartilhamento de conhecimento e o Amaury escreveu excellent tal.
Amaury writing.
Amaury was watching you.
He was
em YouTube.
Alguém mais quer fazer alguma pergunta? Comentário, sugestão? Okay. I have a small. I don't know what was the modeling objective in the project. I cannot understand clearly. For me, it's not. The project is very big,
so one of the underlying idea of the project is to see if, are biogenic volatile organic compounds a significant fraction in all the ozone buildup over the MASP area. So the idea first of the modeling is to couple it to the Megan emission module. Rita, you can complete if you want. And in order to see whether those biogenic emissions play a key role. And also the idea is, because we don't know anything about those biogenic emissions, is to constrain the emission model with the observation we get. Especially we will quantify some emission factors, for instance. So the idea is to try to, let's say, have a biogenic emission model which is much more close to the real world.
Because normally the forecast of ozone or any simulation of ozone, you can capture the diurnal cycle, but in general you cannot capture the high levels and the low levels in the streams.
So the extreme is not well captured?
No. Okay. Normally,
and do you?
I saw many presentations of ozone here in Europe, and this problem is more or less.
I don't know if the project has the ambition to resolve this, but
I don't know.
But I don't know if it corresponds to.
Do you know why?
No, actually, usually the models predict lower levels of ozone during the evening because of high NOx, but usually the extremes aren't very well reproduced. So maybe we can add some isoprene and then see what happens.
I have a question.
I haven't, yeah.
You have a question, but
Okay.
I have also one, but I don't know if it's here or after. Which
Okay.
is-
Thank you, Agnes, one
Thank
time.
you for being here.
Na próxima semana tem mais seminário para o pessoal que veio aqui pela primeira vez. Vai ser um seminário sobre ciclones. Então, a pessoa vai apresentar online, ok? Tem bastante gente, mas eu vou falar aqui.
os nossos olhos, o pescoço, né? A gente vai poder só pegar pelos pescoços.
Cadê o